Okay, next we're going to pick up with some of the methods of protein synthesis, amino acid synthesis. I'm not going to spend a lot of time on this. A lot of these are of historical interest, but we can start, for example, with alpha keto acid, make the amine, and then reduce that. One of the shortcomings, though, is that we get a racemic mixture. This is actually similar, though, to what happens in biological processes. In fact, in biological processes, an alpha keto acid often is converted to an amino acid. Okay, and this is showing that. Okay, using NADH, we have a reductive amination process that occurs. Okay, this one I'm just going to mention. This is an important biological process in which we have an amino group transferred from one amino acid to a oxaloacetic acid. Okay, and enzymes that are used to catalyze these reactions are called transaminases. Earlier we talked about the Helm-Vollard-Zielinski reaction, but again, this gives a racemic mixture. I'm going to skip over the Gabriel synthesis. This is the Strecker synthesis. Again, it's mostly of historical interest. Okay, so we start with acetaldehyde and with ammonia and HCN, we produce an aminopropionyl. Hydrolyzing this, we get a racemic mixture of alanine. Okay, so this goes back to about 1850. Okay, I'm going to move through this quickly. Okay, usually only the L enantiomer is biologically active. And if we do produce a racemic mixture of amino acids, we can convert the amino acid to a salt using a chiral acid or base. And then these diastereomeric salts can be separated by column chromatography or perhaps by recrystallization. We can also use an enzyme such as acylase that will react with only one enantiomer. Since we have a carboxylic acid, we can do esterification of that. This is mainly of interest as a protecting group. And then we can, if we want to regenerate the amino acid, then we can use hydrolysis. Okay, so acylation of the amino group can be achieved with a reagent such as benzyl chloroformate, which forms an N-benzyloxycarbonyl derivative. And again, this is used as a protecting group. And the reason that this particular protecting group is used is because it can be easily removed when it's no longer needed. Okay, this is a method of primarily of analysis of amino acids. If we separate amino acids, say using paper chromatography or thin layer chromatography, okay, we can visualize the amino acids by adding this reagent called nenhydrin. This is typically sprayed onto the plate, and it produces a purple color. So wherever you have a purple spot, that indicates an amino acid. Okay, if you get nenhydrin on your hands, it turns them purple because of the proteins. And this is also used in electrophoresis to help visualize the bands. Okay, amino acids are resonance, the amide group is resonance stabilized, okay, as shown here. And so that makes these 4 atoms coplanar. Amides are very stable in neutral. And this is a simplification of how it occurs, but you have a removal of water here, and these 2 amino acids join together to form a peptide. Okay, polypeptides usually have molecular weights of less than 5,000, and proteins are typically 6,000 to 40 million. Okay, so this is a human hormone called bradykinin. And it is made out of 4 to 10 amino acids. That's an oligopeptide. And it is conventional to draw the peptide structure with the N terminus, okay, over here on the left, and the C terminus on the right. Okay, so the sequence of amino acids is in this order. And if we were to reverse this sequence, we would actually have a different peptide. Okay, so peptides are named from left to right. Many peptides contain disulfide bridges, and formation and breaking of the disulfide bonds is reversible. This is an important feature in the structure of many proteins. Okay, here's human oxytocin. And you can see it has a disulfide bridge here. Okay, here is the C terminus, but it's in the amide form. Here's bovine insulin. Insulin is composed of 2 separate peptide chains. There's the A chain containing 21 amino acid residues, and the B chain that contains 30. Okay, I'm going to go through this fairly quickly, but peptide structure can be determined first. Usually the first step is to cleave the disulfide linkages and then determine the amino acid composition. And then we want to determine the sequence, starting from the N terminus. And then we do a C-terminal residue analysis. Okay, there may be a partial hydrolysis. Okay, so disulfide cleavage is often accomplished by using peroxyformic acid. There are amino acid analyzers, automated analyzers that are available. Okay, so we separate the individual peptide chains, and then boiling with 6 molar HCl is going to break pretty much all of the peptide bonds. And so this separates it into individual amino acids. And then there are various techniques for sequencing. One of them is called the Edmond degradation. This uses phenyl isothiocyanate followed by hydrolysis. And what this does is it removes the N-terminus amino acid. And then you use a phenylthiohydantoin derivative, which is then identified by chromatography. And this is useful for peptides with less than 30 amino acids. Okay, so we remove the amino acids one at a time, and then convert them to this phenylthiohydantoin, which is then identified. Okay, and these will have different characteristics depending on what the R group is. Okay, and then there's also the Sanger method, okay, in which we react with 2,4-dinitrofluorobenzene. This is a nucleophilic aromatic substitution where this replaces the fluorine. And using 6 molar HCl, we break this off, and this is now colored. Now, to sequence from the C-terminus, the enzyme carboxypeptidase cleaves the C-terminal peptide bond. And since different amino acids react at different rates, it's difficult to determine more than the original C-terminal amino acid. Sometimes we can get useful information from partial hydrolysis. Trypsin cleaves the carboxyl group of lysine and arginine. Chymotrypsin cleaves the carboxyl group of phenylalanine, tyrosine, and tryptophan. Okay, and I don't necessarily expect you to remember all of that. Okay, I will go through very briefly how peptides can be synthesized. In solution, unless you're talking about a short peptide, this is pretty inefficient. So there was a method that was developed by Merrifield in which solid phase is used. And I've done a synthesis similar to this with nucleic acids, DNA and RNA. But you start with one of the termini, and this is attached to a polystyrene bead, or it can be a controlled pore glass. The intermediates don't have to be purified, and you use excessive reagents, okay, in order to drive the reaction to completion with a solvent rinse. And then the real advantage is that this can be automated. We can program the sequence that you want, and this is so widely used that it's now become very cheap unless you're using really unusual, unnatural amino acids. But you can program in the sequence you want, and it can be automated. Okay, we can make quite a bit larger peptides than we can from solution phase. Okay, so we start out with by attaching the C terminus to this polystyrene or controlled pore glass bead, and the amino group is protected. And then there's a series of reactions where the protecting group is removed. There you have another amino acid connected, and that one has a protecting group, and then the process repeats itself until we get the chain that we want. And then once our chain is complete, then we can use a method to cleave it from the solid support. Here they're using HF. In the case of DNA and RNA, we used trifluoroacetic acid. I'm going to skip over this. Okay, and then in the next lesson, we'll talk about classifications of proteins and levels of protein.